Welcome to my presentation on enzymes. Enzymes are proteins that speed up chemical reactions in cells and increase the rate of bodily functions. A type of protein made from amino acid and peptide bonds, each enzyme will only help with one reaction. For example, if you were to be lacking in streptokinase, then you'd have a very hard time breaking up blood clots. But if you had too much, then you would have a hard time healing cuts and wounds. Some enzymes break up molecules, and some bind together two molecules. This helps the body to process them easier. The molecules an enzyme works with is called the substrate, and when an enzyme binds on, it binds to a region called the active site. There are two theories about how an enzyme binds onto a substrate. The first is the lock and key model, where an enzyme is shaped specifically to fit the substrate. The second is the induced fit model, where they both adjust to fit into the other. Enzymes are very important for maintaining homeostasis in the body. Competitive inhibitors compete with substrates to bind to the enzyme at the same time. These can be counteracted by increasing the concentrations of the substrate. Uncompetitive inhibitors are when the substrate and the inhibitor bind to the enzyme at the same time, and the binding of the inhibitor plays a factor in the binding of the substrate, and vice versa. Non-competitive inhibitors do not bind to the active site, and instead bind to a different one called the allosteric site. This will stop the enzyme's productivity by changing the shape of the active site. All of these reactions generally do not cause a chemical change and can be reversed through dialysis or dilation. A couple example of enzymes in the body is amylase, which is made in the mouth, and this breaks down starch molecules into sugar molecules. Lipase turns fats and oils into fatty acids and alcohol. Nucleases break down nucleic acids into nucleotides. Pepsin and trypsin both turn proteins into amino acids. Practical uses of enzymes include being in a lot of detergents and other cleaning supplies. These products use enzymes that break down proteins such as trypsin, chymotrypsin, and carboxyl peptidase. Enzymes are also used in biodegradable plastics, so that when the plants they use to create the plastics are processed into plastics, they have the shorter polymer chains which are easier to break down than the longer ones and the more traditional ones. Another use for them is in paper manufacturing, to remove impurities. This helps the mills that produce the paper to not be clogged as easily, and for the paper to be of a better quality. The enzyme used for this process is called esterase, which reduces the ester bonds of the residue of the paper so that it can be removed easier. The article I am using is titled Bioethanol Production Through Simultaneous Saccharification and Fermentation Using an Encapsulated Reconstituted Cell-Free Enzyme System. This article was published by 5 authors at Kyungpook National University in South Korea. The premise of the study is that they use yeast as a substrate in fermentation and saccharification enzymes to ferment the yeast, which have been put into a capsule so that it can be transported easier. The reason they put the yeast solution into capsules was that since the enzyme prospers in temperatures close to 50 degrees Celsius, where the yeast cannot survive, it was previously forced them to run the process at 35 to 37 degrees Celsius, where the enzymes are not nearly as effective. This is demonstrated here, where you can see that as the temperature increases, so the bioethanol production decreases. And you're allowed to have a much higher yield of bioethanol from each temperature when you're using the encapsulated system. Same goes for the pH relationship between the bioethanol. The values are much higher with the encapsulated system than with the bare system. This is also able to be used or reused up to 15 times, with the yield steadily decreasing. They then used the ethanol that the yeast fermentation produced and converted it into biofuel. This is a really important study because it is a new way to optimize this process and one step closer to completely phasing out fossil fuels. This is my bibliography.